this is a protein family, the photolysases or cryptochromes, which had been studied both in bacteria and in plants. In bacteria, the parent protein, the one that had been studied in most detail, was shown to be a blue light-dependent DNA repair enzyme. So this protein actually is involved in repairing UV damage to DNA and functions in a wide range of organisms. And the fact that it exists in E. coli as a repair enzyme and in other higher organisms attests to the fact that the protein has been around a long time doing the same job in these diverse organisms. And it has cousins, that is, related proteins, that function as blue light photoreceptors in plants to carry out a number of developmental functions. And now we even know that these proteins also contribute to circadian light harvesting in plants. And so when the plant data emerged, and when there was even a paper published on the presence of these proteins in humans, we were very interested in exploring the idea of whether these proteins might be the light harvesters, the actual photoreceptors in fruit flies. And we had known for 30 years or more that blue light were the wavelengths that were most effective in phase shifting the circadian clocks in flies. But we had no idea for all this time, well before I became involved in the field, of what was the real molecule, which was the photoreceptor. And so with this possibility that cryptochromes, which were blue light sensitive proteins, might do this job, there appeared, through the magic of the Drosophila Genome Project, just at the time we were thinking about this, there appeared a Drosophila cryptochrome as a sequence about which there was nothing known. So we cloned this gene using the information from the database, and the first experiment we did was to insert this gene into fruit flies, wild-type copies of the gene, extra copies, all over the fly, in all of the fly tissues, just giving the fly a lot more of this protein. And we wanted to ask, would this make the flies more light sensitive? Would it make their circadian system more light sensitive? And what's shown here in blue are the flies which have extra copies of the gene, and what's shown in yellow are the flies which are normal, which have the normal one copy of the gene. And this, what was assayed, was a phase shift at 2 different times of day in response to a light pulse. So normal flies experience a phase delay if one gives them a light pulse at this time of night, and they experience a phase advance if one gives them a light pulse at this time of night. And much to our disappointment, when we took overexpressed the flies with extra cryptochrome genes and did the same experiment, we found little or no effect. And then someone in the lab had the bright idea, no pun intended, that maybe we should assay these flies at lower light intensities, because perhaps when one exposes the flies to an intense shock of light, that the normal amount of cryptochrome was sufficient to gather all the light that was there, and having extra cryptochromes wouldn't make any difference. But if there was limiting light, so you just give low intensity illumination, then the normal flies would be limiting for the amount of light harvesting protein that they had, and the transgenic flies would respond more robustly to this low-level illumination. And that's exactly what happened at low levels of illumination, both at this time of night when there is a delay in response to the light pulse, and at this time of night when there's an advance in response to a light pulse. We saw a very dramatic change in the response, suggesting that cryptochrome really was the light harvesting protein. So remarkably, at the same time, we were screening for new mutants in fruit flies using this strategy of sprinkling this period gene luciferase reporter around the genome and looking for locations in which this would cycle, indicating that the locations where the reporter gene had landed were actually potential clock genes. And one of these locations turned out to be flat in a particular mutant, so that the mutant failed to undergo these oscillations, and it turned out that this mutant was in the same gene, the same cryptochrome gene that we had cloned. And here's an example of this response of the timeless protein to a light pulse. Here's the degradation I showed you previously, where the wild-type timeless protein disappears. And here is the Cry mutation called Cry-B, named by my colleague Jeff Hall after the rock and roll song Cry Baby. And you'll notice that in this mutant, a 15-minute light pulse or even a 1-hour light pulse fails to degrade the timeless protein. So the reporter gene fails to cycle, and the reason that it fails to cycle is because it can't get any light information. The flies are light blind and therefore don't undergo circadian oscillations. So it also turns out, remarkably, that this cryptochrome mutant strain is also resistant to the effects of constant light. So it turns out that all organisms that have been tested need some period of darkness for their circadian clocks to function properly. If you maintain a mouse or a fruit fly in constant light, then these animals' circadian systems respond adversely. They don't like to be in constant light. And two things happen. Under low-intensity illumination, the period alters. So instead of being the natural period of 24 hours or 24.5 hours, the period may lengthen to 27 or 8 hours. And in some organisms, the period actually shortens in response to low-intensity illumination. In response to high-intensity illumination, however, many organisms, including fruit flies, actually go arrhythmic. So the clock actually stops when the animals are exposed to continuous illumination. And yet these mutant flies that have little or no cryptochrome protein, these mutant flies continue to be rhythmic, and only a small portion of them are arrhythmic, and the period of these cryptochrome mutant flies is pretty decent. It's very, very close to the wild-type period. So even the period lengthening doesn't take place. And so I have, I think, next an animation which now illustrates the contribution of both double time and cryptochrome to this cycle, which we're building in a more and more complex fashion. So here's the double time kinase, which puts phosphates on the period protein, and here's the cryptochrome, the light harvesting protein. And so now the cycle starts out as it did before, but we see that the double time kinase, this casein kinase 1 epsilon, is actually the agent which contributes to the degradation of the period protein, which kills the pink proteins, if you will. And if the period protein which is produced gets together with its partner timeless and forms a heterodimer, then the heterodimer is resistant to the effects of the double time kinase. So that's really the distinction between the protein which is degraded and the protein which is accumulated. Then the heterodimers get together and connect, and here is the conversion of cryptochrome from an inactive to an active form, and it goes and kills timeless. And then the double time kinase goes and kills the per proteins, turns over the proteins, and transcription becomes anew. So this has added two elements to the story. First, the double time kinase is actually the agent which turns over the period protein or assigns the protein as being a substrate for degradation in the manner that I referred to previously. And second, cryptochrome is actually the light harvesting protein, and cryptochrome, when it's converted from an inactive to an active form by light, cryptochrome actually then leads to that very rapid degradation of the timeless component, which then begins the day anew leads to the turning over of the period protein and starts the cycle once again. So this is an indication, this gives you a flavor for really a piece of biochemical data which shows how a double-time mutant actually changes the molecular profiles of these proteins. So here is the wild-type protein profile I showed you previously. This is the timeless protein, which accumulates over the course of the night. And this is the period protein, which also accumulates over the course of the night, and then in the morning experiences this slow disappearance, this degradation phase. And here is a double-time mutant strain, actually a heterozygous strain. This is an allele, a mutant that we uncovered in our screen in our laboratory. And this strain has a long period. It has a period of 27 or 28 hours instead of 24 hours. And note the fact that in the morning, this protein is disappearing with kinetics which take much, much longer than the wild-type protein. So the wild-type protein is effectively gone 5 to 7 hours after the lights have turned on, whereas this protein hangs around for a long time. It's ineffectively degraded, and as a consequence, the whole profile is extended, and the 24-hour cycle actually takes 27 or 28 hours. Now, if I might have the next slide, please. Oh, I think we go now to the animation. So this now gives you a flavor for how the double-time mutant protein, this mutant kinase, which doesn't work very well, compares with its wild-type counterpart and the effect of that mutant on the cycle. So here's the double-time kinase destroying the period protein, and notice that it's having trouble functioning. It takes a couple of shots to kill the pink protein as compared to the normal one shot which the wild-type protein is able to affect. Yet the accumulation in the cytoplasm in flies occurs pretty normally. And so these heterodimers accumulate in fairly normal fashion. They migrate into the nucleus. They make contact with the positive transcription factors, extinguish transcription, and now here's the conversion of cryptochrome to the active form, and now watch what happens. Timeless disappears very quickly, and now the nuclear form of the kinase starts to degrade per, and the mutant form has trouble keeping up. It's working more slowly. This takes longer to go away, and as a consequence, the turning on of transcription the next day occurs more slowly in the mutant strain, the strain with the mutant double-time kinase, as compared to the wild-type strain. So that's actually a fairly realistic depiction of how that mutant actually lengthens the protein. So now I'll just say a word about output genes. I should say that there are a couple that we know about, vril and takeout, which I won't have time to talk about. One of these is a transcription factor, and one of these actually connects to food and feeding behavior. But I will mention the pigment dispersing factor, this hormone that's important for locomotor activity rhythms. And we discussed this briefly yesterday, this protein. I told you that this neuropeptide is present only in 8 cells, and those are the 8 neurons in the brain which have very prominent, very intense, very high levels of the period and timeless proteins. And here's a picture of these 8 cells which are stained with a fusion protein of PDF. And these 8 cells are stained, and also their processes are stained. And these cells make contact with the eye. They also make contact to the same 8 cells on the opposite side of the brain, and they make connections up here to interneurons, which appear to be important for the locomotor activity program. So this neuropeptide is important for locomotor activity rhythms, but it also represents a tool that we can use to ask and answer an important question. We can take advantage of technology that was developed over this past decade by Norbert Perimon and his colleagues, and we can take the control region of this PDF gene and we can use it to express a transcription factor only in the cells in which that control region is programmed to express. And I told you that that gene is only expressed in those 8 cells important for locomotor activities. This then allows us to express the cryptochrome light harvesting protein only in those 8 cells by restricting its expression using this particular technology developed for fruit flies. And if we express this cryptochrome protein only in these 8 cells, we can now ask if the effects of the mutant, the strain which is missing the cryptochrome protein, is rescued by having this protein only expressed in those 8 cells. And it turns out that all of the locomotor activity defects that exist in the strain, in the mutant cryptochrome strain, are rescued by expressing this protein only in those 8 cells. So this allows us to draw an important conclusion, and that is that these 8 cells, these lateral neurons, as they're called, or pacemaker cells, are really the fruit fly equivalents of the suprachiasmatic nucleus as far as locomotor activity is concerned. Because all the elements that we need to drive a clock, both input elements, the cryptochrome protein, the 5 pacemaker components, the clock, cycle, per, tim, and double time, are present in these cells, and even an output component, a neuropeptide, which is sent down the axon and which connects to the next motor centers, are present in these 8 cells. And if these elements are functioning in these 8 cells, then we have a viable clock. And of course, the fact that we can rescue light harvesting by only expressing this protein in these 8 cells tells you that these brain cells are directly photosensitive. So they connect directly to light, and these represent then deep brain photoreceptors, and really the first example of this kind. Of course, you'll appreciate, and something to think about both for questions and in general, is the fact that our skull, of course, prevents a comparable biological solution to the problem for mammals. So this is a contemporary view, a pretty sophisticated contemporary view of the clock system in flies. And so this brings me to a summary where I have told you how these 5 proteins are arranged in this feedback loop that generates 24-hour oscillations. Cryptochrome is the light harvesting protein which connects to timeless, which is the light sensitive, the light relevant central clock component. And in work I just didn't have time to explore in any detail at all, I'll simply tell you that there are output genes, genes which connect directly to behavior, which are related to this transcriptional feedback loop in a very similar way. They're regulated by the same sets of genes, and they exhibit 24-hour periodicity in their gene expression, and that connects to behavior. So with that, I'd like to thank my lab members, the people in my lab who are responsible for this work. I'd like to also thank a lot of ex-lab members, as well as my good colleague, friend and collaborator, Jeff Hall, who really brought me into this field 18 years ago and has taught me an incalculable amount of work, amount of information about biology. And finally, I'll leave you with just 3 brief sentiments. First of all, I hope I've given you some feeling for the remarkable progress that the field has made in understanding these timing mechanisms in the brains of all animals and the relationships between clocks in flies and other organisms like ourselves. Secondly, the importance that genetics has played, both in general and in Drosophila, as an entree into a biological problem which otherwise turns out to be quite mysterious, if not really opaque. And last but not least, how much fun you can have in pursuing your dream in this kind of an enterprise. Thank you very much. So, a question from the House. Wow.
Have you tried substituting heat for light, or maybe different combinations of darkness but with a warmer temperature?
So that's a great question. We've really not done that in fruit flies much. It's been done in other animals, and in particular in Neurospora by Jay Dunlap and his colleagues. And it turns out that you really can try to play off heat and light and ask which is more important. It's tricky because it's not completely clear how to play with the intensities to try to get a sense of who plays the dominant role. Our sense is that in flies, light is more important, the temperature, but that's a very soft conclusion because really the proper experiments haven't been done yet. Yes?
How does the circadian clock sustain the 24-hour cycle in the absence of light, which is necessary in the feedback mechanisms of the per and Tim cycle?
So it appears, for reasons which are not completely clear, that these oscillators, at least in their central locations, the oscillators in those 8 cells as well as in the brain of mammals in the suprachiasmatic nucleus, can keep ticking away without any external influence. And that distinguishes them from the oscillators which are present elsewhere. And it's not completely clear what that distinction is. There's a question from Miami. Go ahead, Miami.
Hello, Doctor. I'm Julian M. Moranci from Miami Northwestern Senior High School. And what I would like to know, Doctor, is do the pacemaker genes that you mentioned in the fruit flies have a similar DNA sequence to the pacemaker genes of a mammal?
So that's a very good question, and the answer is they have a very similar DNA sequence, not an identical DNA sequence. We believe that most of those differences are due to what are called neutral mutations, which don't have very much impact on what the gene does, that just represent the drift of the DNA as fruit flies and mammals have separated. But it could be that some of them really are functionally distinct and affect the gene's function in subtle ways. So I'm told that it's time to go, and I want to thank you all very much, both here and at these remote locations, for your attention. Thanks again.
Thanks, Michael. You talked a lot about feedback, so now let me give you some feedback. That was a really terrific lecture, and I particularly liked the way that you showed how early models were not wrong, but they were just incomplete, and then latter data allowed you to get a more complete model, something that happens so often in science. We're going to take a break now for half an hour. When we return, Joe Takahashi is going to take us from the fruit fly to the mammals and tell us about how mouse genetics and genomic databases have allowed us to learn more about circadian rhythms in complex creatures. He'll also tell us a bit about how this research might be of use to human patients. See you then.